introduction. So welcome. Welcome to Science on Tap. It's brought to you by the Laboratories Bradbury Science Museum, the Los Alamos Creative District, and tonight the wonderful drink and refreshments are being sponsored by the local chapter of the American Chemical Society. These talks are held every third Thursday at 5:30 here at Uncorked. And my name is Linda Anderman, and I actually handle communications for the Bradbury. Feel free to get something to eat during the talk. I'm sure our speaker won't mind. Just as a heads up, our next session is going to be on June 15th, and it's going to be Jeff Reeves, and he's going to be talking about space weather and how it affects us here on Earth. If you need more, here we go. This is not a lecture, and it's not a formal presentation. It's a gathering of people interested in cool science topics and to share our knowledge and interest. The format is basically, our speaker will get up and talk for about 15 minutes or so, and then afterward there will be a discussion that everyone can engage in, and we'll kind of keep going until we either tire our speaker out or we run out of time. I'll moderate as needed. We should go until about 6:40 or so. So about tonight's speaker. So Patrick is a I need to have my glasses for this part. He has smaller print than I did. Patrick Gazda has been a postdoc working with Roger Weens, who is a principal investigator for ChemCam, which is part of the Mars rover that's up on the planet right now. He has a PhD in geology from the University of Hawaii and a Bachelor in Science in Chemistry from Ursinus College in Pennsylvania. His research topics include planetary science, specifically Mars, how convenient for his line of work, meteorites and Europa, astrobiology, and prebiotic chemistry. So, with no further ado, let's give a hand to Patrick Gazda.
All right, thanks for the introduction. Am I speaking loud enough for everyone to hear, I hope? Okay, cool. So, like Linda said, I've been a postdoc here for about 2 years, and I work on the ChemCam instrument, which is an instrument that's on board the Mars Curiosity rover, which is currently exploring this part of the planet called Gale Crater. In my recent work, I've been using ChemCam data to detect boron, which is an element, it's the 5th element, on Mars for the first time. And before I really get into talking about this boron measurement on Mars, I'm going to talk to you about astrobiology and prebiotic chemistry. So this is basically the field of basically how you make life out of basic chemicals, from basically making life from scratch. And so this will explain why measuring boron is important. So astrobiology, this is this really wide-ranging field, but essentially what we're trying to do is answer 2 main questions, which is where is life in the universe and how does it form? So generally we know about how life forms based on, there's just this simple multi-step process. You have geology, geological processes that then make chemical processes, and then that forms biology. Simple. So kind of the gist of this is that first you form a planet, and then, you form a planet and you have like geological processes that occur. So you're making oceans and the atmosphere, and the water is altering the rocks and doing lots of interesting chemical things, and then you're forming organic molecules. These organic molecules then start to interact with each other, and there are inputs from geology. So like, what was the temperature of the water, and what ions are in the water that influence what those chemicals do. And then eventually these chemical systems start to interact with each other, and you form some kind of proto-life. And this proto-life, there's one hypothesis called the RNA hypothesis. So you have this RNA world. So RNA is this kind of interesting molecule. It holds genetic information, but it also can evolve, but it also can reproduce. And so after a long period of time, you have a lot of evolution of this RNA, and you could start to form life as we know it, so cellular life. And so prebiotic chemistry is essentially the study of all of the details and the mechanics of how those organics are forming on the early Earth to forming proto-life and then eventually life as we know. So to understand these details, you really need to understand something about geology. So the geology of the early Earth is going to tell you about the conditions that were going on at that time. So again, like what was the temperature and what was the atmosphere made of? So in principle, you could take some rocks from the surface of Earth or Mars, or this, we do this on Mars as well. You take the rocks, you go into the lab, you study its chemical composition, and then you also study its isotopes, and that would tell you how that rock was formed and what's its history. So it tells you the conditions of, say, the atmosphere. But you can't really do that for the early Earth, unfortunately, because there's really no rocks from that time. We're talking about having rocks survive on the surface of the Earth for like 4.5 billion years. So, you know, Earth has plate tectonics, and that can just destroy rocks completely. And then you have water, which can dissolve the rocks away. And then you have life, which is super annoying because the rocks will still be there.
Yeah.
The rocks will be there, but it'll change like the isotopic composition and completely erase like any history, like any of the signature of the history. So this is where Mars can come in. Okay, so Mars is dry, probably has no life on it, and the surface of Mars is older than 3 billion years. So you can go to Mars and study those rocks and study ancient rocks. We also know that Mars probably had a similar history as like the Earth. So there's a lot of water on Mars, and it changed the rock's surface. And so the idea is you can study Mars to sort of start to understand like early, you know, how planets evolve in like early stages. And then we can, if we found life on Mars, you can compare that to Earth and try to figure out, you can tease out like, what are the necessary components for life? Okay, so you're like, why are you talking about boron now, like with this whole origin of life story? So, kind of the spoiler here is that boron can play a vital role in the production of RNA. So, let's talk about RNA kind of in more detail. So again, you know, RNA is this interesting molecule. It's in all types of life forms. So like everyone in this room and the plants and insects and bacteria all have RNA molecules in their cells. And again, RNA, you know, holds some genetic information as well as it can kind of reproduce itself. But it's not really life as we would call it life. So RNA, what does that stand for? It's ribonucleic acid. Its basic constituents are these things called bases. This is what is the genetic information. And it's held together by phosphate and ribose. Ribose is a type of sugar. RNA is actually not very easy to make, even in the lab setting, from like the basic materials. And if you think about it, like back in the time when life is being formed, which is like 4.3 billion years ago, it's a very messy system. You have all these organics in there that shouldn't be there. You have water and ions dissolved in the water, and it's a super messy system. So it's actually not trivial to make RNA by itself. And then you have ribose, which is the sugar. It's a very important part. It's very unstable. So if you have ribose or any type of sugar and you put it in water and just let it sit around, it'll just decompose. And this is an irreversible reaction. So it just destroys all the ribose, and you can't make RNA. Secondly, when you're trying to make ribose out of like basic materials that might be on the early Earth, it's a runaway reaction. So it forms RNA, but it gets quickly consumed, and it makes these more and more complicated molecules, and eventually you're left with like this brown crap at the bottom of your flask. That's not very useful for making RNA. So it turns out other people, not me, have discovered that if you take boron and you put it into water, it actually will react with ribose to form a stable compound. So that's cool. So now you can actually